I just sent you an email about this. If you go to this URL, you'll find something that says student post-test. Don't try to take the pre-test. It's all shut off. All right, so you only get the post-test. And the student materials evaluation. If you do either of those, you will get 1 point for each. All right, I'd actually prefer if you do both. It's completely voluntary. You don't have to do it. I am doing some research on this. We're trying to get this module buffed up and improved. And hopefully we're going to publish it because you might have realized everything we used is accessible to everybody in the world. So I'm trying to show instructors elsewhere how they can do this great module, even if they don't lucky enough to have labs and equipment and mice and all that other good stuff that we tend to have at UCLA. So I'm trying to get it in some kind of shape to send it out to the world and show that it indeed is an effective teaching tool. So if you do that, I'd appreciate it. If you still want, I still have to make this disclaimer, if you still want the extra credit and you don't want to participate, come and talk to me about doing an alternate assignment, which will probably be a one-page paper for each point of extra credit. I have to do this. It's part of the rules. Okay, so that's my pre-lecture, and so let's get on with the real lecture. What I'm going to do in this lecture is I am going to go over what I wanted you to get out of this unit, and hopefully that you did get out of this unit or will be getting shortly out of this unit. So my pedagogical objectives, my teaching objectives, first of all, was to give you a short review of genetics, which should indeed have been a review for all of you. We talked a little bit about genes being linked and linkage, and if genes were linked on the same chromosome, we talked about the fact that we would then predict that we would get a 50-50 inheritance pattern if they were indeed linked. And then we also talked about the fact that that 50-50 prediction doesn't hold up, that it's not quite 50-50, and that's how people discovered recombination, okay? And we also talked about how our recombinant inbred strains came to be. And since I got some questions in lab, I'm going to go over this slide in some detail. So our recombinant inbred strains started out with two strains that were fully inbred. These are the F0s. So we either had the C57BL/6Js, which we, from that point forward, started calling the B mouse, or we had the DBA/2Js, which they're called the D mouse. So these are our F0s, and they had to be highly discrepant on our phenotype of interest. And I checked it out, and indeed these two strains were really discrepant on the size of their olfactory bulbs. All right, so that makes them good candidates for this kind of analysis. So what we did is we crossed these two F0 strains, and what we're representing here is, with these blue or red bars, is a single chromosome pair. But this would be, what you're going to see in this slide would be true for all the chromosome pairs. So, of course, in the F1 generation, these particular mice, these mice are isogenic. They all have the same genes, but with regard to where their genes came from, they're completely heterozygous, right? So they have one chromosome from their mom and one chromosome from their dad. Each one of them has this kind of pattern of chromosomes. One from the mom's strain, the B strain, one from the dad's strain, the D strain. What we then did was we crossed these F1s. And when we cross these F1 strains, what happens is we get recombination. Of course, you always get recombination, but in this case, it's going to really matter. So when these F1s go to make their gametes, these chromosomes from the mom and the dad are going to get back together. There's going to be some crossing over and some recombination. So in that fashion, we are going to then be mixing up the DNA from the mom strain and the dad's F0 strain. So it's going to be mixed up. As you can see, these chromosomes now carry kind of a collage of the DNA from the F0 strains. And as you can also see in this diagram, these particular, again, at this point, a particular strain could be heterozygous for a given gene. So let's just take this first character right here, and we see that this individual, let's say that I'm putting my light on a region that would have a gene. Would that gene be heterozygous or homozygous? Hetero, okay. So we don't want that. We want to mix up the F0 DNA in some fashion, but we want everyone to be fairly uniform. All right, and so the goal to get them, how are we going to get all these F2s to be uniform? It's not a rhetorical question. Inbreed them. We're going to inbreed the heck out of them for 20 generations, brother-sister pairs for 20 generations. And what that will do is a given gene will lock into being homozygous. So a given gene will have the same allele. We will still, at the end of 20 generations, have some kind of combination of the F0 DNA. Okay? But, and let me go on to say, every individual within that recombinant inbred strain will have essentially the same DNA. Of course, with some asides, like male and female, obviously they can't have perfectly the same. They'd have X's and Y's, would be different, but all the other chromosomes should be pretty much the same. Okay? So within a recombinant inbred strain, those chromosomes are the same, but across these recombinant inbred strains, they're going to represent different combinations of that F0 DNA. What we're then going to do is say, okay, so now we're going to see how these different combinations of the F0 DNA are going to have, will they have differential phenotypes back to our olfactory bulbs? Will their olfactory bulbs look different across these different recombinant inbred strains? And if they do, we might be able to nail, in fact we were able to probably nail the pieces of chromosome that made a difference in that phenotype. Okay, so along those genes, what we have are markers, or as they are also called, loci. And each of these loci are markers, and there are a whole lot of them across each chromosome. At each of these loci, they've already identified if that locus at a given locus, does that recombinant inbred strain have the B form of the DNA or the D form of the DNA. Okay, so what they've done is they've done PCRs on little, this in fact is a PCR from a little microsatellite, just a tiny stretch of DNA. And this ought to look pretty familiar to you from the Bolter Line Master unit. Here's the molecular weight ladder, here's the loading wells, and this was the B, F0Bs, here's the F0Ds, and all these others were the recombinant inbred strain, different recombinant inbred strains. And this is the very first F2 generation, okay? So some of these you can already see at this given locus have already settled in the B or the D form, all right? And so they're homozygous for that B or D form. And some of these are heterozygous at this point, but this is the first F2 strain. Okay, so if these F2s kept getting inbred at the end of 20 generations, what would you expect this banding pattern to look like? They're either the Bs or the Ds, every last one of them, okay? Are they all going to be Bs? No. Some are going to go B, some are going to go D, all right? Now, think with me for a moment. Let's say we find, let's go back to this. Let's say we find, now here's our highly inbred strains. Let's say we have a marker, which would be just a tiny, tiny, tiny piece of chromosome. And it is for this one, at the tips here, it's the B, all right? Would you expect, irrespective of this diagram, would you expect the genes around there to be from the B or the D strain, and why? B, okay, because of, because they're B. Well, you expect them to be linked, right? Because this recombination, as you can see, tends to take place in big hunks, all right? It's not like we're getting a scramble every 10 bases or something like that. It's big hunks. So very likely, if you have a B marker, the genes around there came from the B strain. If you had a D marker, the genes around there came from the D strain, all right? A tiny assumption, but it's probably right. Okay, we talked also a little bit about qualitative versus quantitative traits. I think I asked you about this on the exam. Qualitative traits, did you guys have to do fly labs when you did genetics? Oh, you're so lucky. Otherwise, you had to get up at 4 o'clock in the morning, make sure they're all virgins and all this other stuff. It's terrible. But anyway, lucky you didn't have to do all this, but you did probably talk about all these traits in flies, right? So red and white eyes and wrinkled versus straight wings and all this stuff. Those are qualitative traits. They're probably influenced by a single gene. They tend to follow simple patterns of inheritance. Phenotypes can be characterized in a nominal scale, so they either have red or white eyes, something like that. And usually the trait expression is typically unaffected by the environment. What we're looking at here with this QTL analysis is something that's most of the traits in the world, and those are quantitative traits. And these tend to be influenced by multiple genes, perhaps interacting, don't follow simple patterns of inheritance. Phenotypes are measured on a continuous, that is to say an interval scale. I say interval scale, that makes sense to all of you? You took statistics, you told me. Yes, all right, so things that, at least have equal intervals across the different numbers. And the trait expression may be affected by the environment. So most traits that you think about in terms of human traits or even mouse traits are quantitative rather than qualitative traits. Okay, so again, one of my first objectives was a small review of genetics. And then one of my other big objectives for this unit was a review or introduction of statistical analyses and concepts. So let's talk about what we saw with regard to statistics. All right, one of the big deal things that we got into here with my buddy Sir Francis Galton is we talked about regression, okay? And we talked about variance, and we talked about the fact that you could take variance and partition it into its various components, all right? And what we were wanting to do was to control for the effects of sex and body weight and brain weight and age, even though we had a set of mice where we had both males and females, all kinds of different body weights, all kinds of different brain weights, huge range of ages. But we thought, oh, these variables could matter with regard to the thing we're really interested in measuring, and we don't want them to influence it. So what we're going to do is go in and control for that. And what did we do to control for that? What was the name of the statistic? Say it loud. Multiple regression. Thank you. Okay, so we went in, and the idea behind that is we could go in and we could take out the variance that's due to sex and take out the variance that's due to body weight and take out the variance that's due to brain weight and take out the variance that's due to age and distill those numbers down so hopefully at the end we would have the variance that's unique to olfactory bulbs, which is what we were measuring, and of course the ubiquitous error that you never quite get rid of everything. All right, so that was the idea behind this. And Sir Francis is back to remind me to say that what we did is we removed the variance successively using multiple regression, and hopefully at the end we had this variance, which was the residual variance that only pertained to olfactory bulbs. Okay, and as we made our way there to talking about multiple regression, we first talked about the case of simple linear regression. And so we talked about the fact that if we had a scatter plot like this here, we could draw a best fit line through that or a regression line through that. What we're trying to do with this scatter plot is predict a y value from an x value. So we want to find the line that best fits and minimizes the squared deviations in the y value. Okay? This all comes back to you? Great. All right, so that's the best fit line or the regression line or what's also called the least squares line. And so in our case, we talked about the fact here we have olfactory bulb volume and body weight, and we want to get rid of the variance that's due to body weight. So we also talked about the fact if this variable body weight predicted nothing about olfactory bulb volume, let's just pretend it didn't, then what would be the best predictor, the best y value that you could have? The mean, right. Okay, so if this doesn't predict anything, then the best predictor is the mean of y. So we're going to see if our variable did any better than just the mean of y. And so our regression line is here. The predicted y's then are the y hats. Okay? And I particularly picked this little point here, happy face point, to show how this works. So we can see that actually we did have a relationship between the two variables because the line isn't flat. And we can also see that we did a little bit better on happy face point than just picking the mean. In fact, so here's the whole variation of happy face away from the mean, and this much of it, of this variation, was predicted by body weight. Okay, does that make sense? All right. So that's great. So what we're going to do is we're going to go through this plot and do this for every last point, grab that variation that's due to body weight and haul it out, and thereby control for body weight. Okay? And take the influence of body weight out. Of course, we didn't predict, not everything was predicted by body weight. That which is not predicted for this particular point contributes to the residual. Okay? And for us, the residual is what's valuable because hopefully our variance that's due just to olfactory bulbs is going to be in that term. Okay, I'm going to put this slide back up to remind you, usually one uses regression to predict something, and the residual is the trash. For us, it's going to be the treasure, all right, but usually the residual is the trash. And I also put this slide up to talk about a point once more that I talked about in the lab, and that is the square of the correlation coefficient. The square of the correlation coefficient tells you what? Right, or by the predictor variable. Right. Excellent. So that tells you the proportion of variance that's accounted for, the square of the correlation coefficient. That's one of the things I definitely want you to take home from this little exercise. Okay. All right. So Sir Francis is going to tell us the variance left over after the variance of the other variables has been removed is the residual variance. It's precious to us because it has the variance that's specific to the olfactory bulbs. By the way, we also talked about the fact that, yes, you can statistically control for things as we had to do in our study, but is it all, why don't you always do that? If it's that easy, just, crunch some numbers, why don't you always do that? Why do you bother to construct really careful control groups? Let's go back and look. Okay, so back here at the old conceptual equation, there is an assumption, and the assumption is that each of these components is independent, right? If that's not true, is this going to work real well? No. Okay, so that's one reason why you want to go ahead and have, if you can, careful control groups. We let a lot of things vary in this study, or the folks that made the mouse brain library for us let a lot of things vary because they wanted to look at lots of different questions. So it's not a cure-all, but a lot of times in the clinical setting, for example, you have people come at you in all kinds of shapes and sizes and genders and whatevers, and you can't control for that, and you might want to factor that out. You can do something like multiple regression to control for that. All right. Okay, so we used multiple regression to statistically control for those variables. We also talked about mistakes that can be made with statistics. They come in two big flavors. We talked about type 2 errors, which are false negatives, and we talked about how to go about getting these false negatives. Of course, you don't want that, but how is it that you can end up with false negatives? This is when you should say there's a relationship or should say there's an effect, and you don't find one. Okay? So if you have a small effect size, you'll make one of these type 2 or beta errors. So in our instance, if we have genes that account for a small proportion of the variance, we may miss them, okay, because their effect size just isn't big enough for us to detect. Another problem, small n. If our sample of recombinant inbred strains wasn't big enough, we can miss some. All right? And the thing that for us was probably the biggest problem is the variance in scores. The more variance you have in scores, the harder it is to get a significant relationship. And that also means especially error variance. If your error variance is high, then you're going to be making more type 2 errors. So let's just pause for a moment and think about that. So we were trying to replicate a study, Williams et al., which is in your reader if you haven't found it yet. And the way that they quantified olfactory bulbs is they had a technician with a razor blade pop it off and pop it on a scale. One technician, one razor blade. Okay? How we quantified the olfactory bulbs is we had all of you take different mice, use that NIH image, and take the successive sections, draw it. Each of you being wonderfully unique individuals as you are, you're all doing it a slightly different way. Okay? So, who do you think is going to have more variation in scores? Us. What's that going to mean for our data relative to theirs? Do you think we will find more or fewer relationships than they found? Fewer. Okay, so let's translate that into QTL peaks, which we'll be talking about shortly. Do you think we're going to get more or fewer QTL peaks than they found? Probably fewer, okay? Because our error variance, I'm willing to bet, was a lot higher just because of how we did it. Okay, while I'm on the topic of how to make mistake with statistics, there's also another big flavor called type 1 errors, and these are false positives. This is when you say there's a relationship or an effect and there really wasn't. So we know that this can happen, so we guard against it by trying to make our outcomes have a lot of stringency before we'll say they're significant. So, and as I'm going to talk about shortly, we did that likelihood ratio statistic over and over and over and over and over and over and over again, thousands of times across the whole genome. Okay, if you do that, that type 1 error compounds. All right, so what we've got to do is make each comparison very, very stringent. Otherwise, we're going to be making a lot of type 1 errors, get a lot of false positives. So I figured it out, and for each point, way back when there were only a few hundred markers, it was like you had to be 0.00006 for each comparison to get to a genome-wide alpha 05. It's probably a lot less than that now. And we also have the suggested rate, which is for each individual point P008, and that would leave us actually with a fair size type 1 error rate for the whole genome, 0.63. But again, QTL is a bit of a fishing expedition, so I decided we'll just live with that. Okay. So, as I already just said, lots of error variance will give us false negatives. Type 2 errors when we do QTL analysis, we probably had lots of error variance. We also, in our tour of statistics, talked about the likelihood ratio statistic, which I'll detail a little bit later in this lecture, and I've talked about in another lecture. Basically, it's a ratio of the variance of all the points to the variance of each of the points around their own mean added together. So it's a ratio of variances. And we'll come back and talk about what the data looked like when it's high or low at a later point. When we got in and were getting ready to do the QTL analysis, what was going to be significant or suggestive was actually was determined by something that's called a permutation test. And these things are becoming increasingly more popular in biology because now we have computers that are pretty fast and powerful. So what we can do is we can use our own data to generate something like a null hypothesis distribution. Did you guys, when you took statistics, study null hypothesis distributions? So what this did was this took the list of markers across the genome and took your values and then randomly assigned your values to a given marker, ran the likelihood ratio statistic for all those markers, and then logged what was the top one, and did that over and over and over and over and over again 1,000 times. Okay, so now what you can see is what the outcome would have looked like with just random data, all right? So from that, you can figure out, okay, what are outcomes that only happen 5% of the time with random data, okay? And so that would be the significant one. And you could also figure out, okay, what's the suggestive level? So what that's going to determine are our cut lines for what is significant and what was suggestive when we did our QTL analysis. Okay? So that came out of this distribution here. Not based on, say, the underlying assumption of a normal distribution, things like that that you probably heard about when you took stats. Okay, so, and this is what a QTL analysis looked like. This is not your data. This is the data from another quarter, but you can see, as I was mentioning, so that permutation tests determine where this red line for significance would fall, and this suggestive line would fall too. And so what we're going to say is any peaks that get over this line here are suggestive, and if we're lucky enough to get any over that line, that would be significant QTL. Did we get any over there, by the way? No, bad news for us. But it's still a happy day. All right. Briefly, in our tour of statistics, and something we will come back to in this course, is an analysis of variance. When we do Dr. Knowlton's unit at the end, we'll also be doing an analysis of variance. Analysis of variance is good for finding the differences among means for several groups. And one of the things that we are interested in is once we cleaned up our data, were there differences among the recombinant inbred strains and the F0s? If there weren't, game over. Then it's probably not heritable, and we can just go home. So now that you guys know how to read one of these tables, don't embarrass me now. All right, so did we get a significant effect of strain? Why do you tell me these things? What's less than 0.05? This one? This one? Which one? That one? Strain. All right. So we got a significant effect of strain. So that suggests that indeed, yes, our residuals are different, that the genotype is going to matter, and we actually also, out of this analysis, got a little bit of a heritability estimate, so about 0.41-ish, okay, was our heritability estimate, and that's good news, because we need at least 10% for heritability for the QTL to work. So that's what we got out of that. Okay, one of my other goals for this unit was to introduce QTL analysis, and I'm not kidding you when I tell you just about every talk I go to anymore has a QTL analysis in it. Since I last lectured to you, I went to a talk and they were doing QTLs on chronic fatigue syndrome. Another guy was doing QTLs on pain sensitivity. So just about anything that has a genetic basis, people are now doing this QTL analysis, trying to figure out what aspects of chromosomes may be contributing to a phenotype, and if you know that, then you can zoom in and figure out what genes in that hunk of chromosome might be contributing. So there's a lot of this going on. It's a technique that you're going to be seeing probably from the rest of your life, and it's getting more and more refined. So I wanted you to know about it. QTL analysis, as I said, is good for detecting the approximate locus of multiple genes affecting the phenotype. We got, I think, 3 peaks, right, in our data, 2 on 6 and 1 on 2. And so that's across all the finding the locus of multiple genes affecting the phenotype across the whole genotype, except not Y. And I have asked you before, and I ask you now again, why not? Y doesn't recombine, so you can't really use this analysis to look at the Y chromosome. Okay. We've already talked about the fact that we were using recombinant inbred strains and that there were markers on, and so what somebody has done to emphasize this again is for every last one of those thousands of markers across the whole genome, somebody has done the PCR reaction and figured out, does that recombinant inbred strain have the B marker or the D marker? So there's a lot of molecular biologists with sore thumbs who've already done this work for us. It's already in the databases, so we can use it. Already been done. So we already know, does that recombinant inbred strain have the B form or the D form? Again, it uses the likelihood ratio statistic, and now let's talk a little bit about what the likelihood ratio statistic does again. And what the likelihood ratio statistic does is, first of all, at a given locus or marker, it takes all of the animals that have the B form and puts them in one pile, and all that have the D form of the DNA puts them in the other pile, all right? Then what it does is looks at the phenotype, or in our case, the residual. So back in the numerator of this thing, it looks at every value minus the mean of all and squares them up. So what it's going to do is take every value minus the mean and then square that value. That's the numerator, okay? And then the denominator, it divides it up with regard to groups. So it says, okay, all those in the B group, we're going to take each value minus the mean of the B's and square that. And all those in the D group, we're going to take each of those values minus the mean of the D's and square that. So if the data looked like that, in this particular example, and we had this thing to grind through, would this value end up being large or small? Large, okay. So my point is, when you get the data to look like this, that everybody who's got the B form of the marker, their phenotype is highly discrepant from those that have the D form of the marker, that's when the likelihood ratio statistic is high. That's when we'll be getting those peaks in our QTL analysis. And that's when, and what that suggests is around that marker, or even at that marker, there are genes that might have had a big impact on that phenotype. In contrast, if it goes in, let's say it goes to the next marker and separates all those that happen to have the B form from all those that happen to have the D form, and you get this kind of a spread in the residual. So you can see that everybody here with the B form, the variance away from the grand mean, the mean of everybody, and the mean of them is pretty much the same, and the variance of everybody around the grand mean and the mean of this marker is going to be pretty much the same. Is your statistic then going to be high or low? Low. Okay, so here you can see just at a glance the phenotype is not highly discrepant, okay? And so then we'll get a place where the likelihood ratio statistic is low. And would you expect there to be genes around this marker that would influence the phenotype? No. All right. So that's what QTL analysis does. And again, this is not your data. It's pretty close to your data. We typically, when we've done this, have gotten a peak on chromosome 6. I think you guys found 2 little peaks on chromosome 6. So again, what it's doing is it's finding the likelihood ratio statistic as a function of all those different markers, lots of different markers across the whole genotype, and doing that likelihood ratio statistic at every last one of those markers. So this blue line here represents the likelihood ratio statistic as it's changing as a function of the markers that are on, and these numbers represent the chromosomes laid end to end to end to end to end to end to end. And by the way, I don't think I've mentioned, but it does pop up in your reading, there's a standard way to array the chromosomes, and the left-hand side is always called the proximal, and the right-hand side is always called the distal. So in your readings, that Williams et al., they'll talk about things being on the proximal or distal end of chromosome 6. That means on the left-hand or right-hand way, but there's a standard way that chromosomes are laid out when people do this and talk about it. Okay? Okay, so squiggly blue line is the likelihood ratio statistic, and this line here is the suggested line, and this line up there is the criteria, is the significant line. Again, we only got things that were suggested, but at this juncture, as I speak to you today, QTL is basically a fishing expedition, so I don't feel real bad as long as we got something that's in the suggested range. If I were in a real lab, I'd go and look under those peaks and see what was there. I wouldn't just say, oh, wasted my time. I'd go look, and I'm sure people do. Actually, I've seen things reported in the literature. Matter of fact, I think it was your paper, Rick, where you did talk about a suggested marker, right? Of course, yes. All right. This, by the way, isn't your data, but it looks pretty similar. Again, we zoomed in on chromosome 6, And we talked about the fact that we wanted to go look in especially places where these lines here represent the SNP density. The more lines, the more SNPs. Would you want to go and look in places where the SNP density is high or low? Why? You got to yell at me. I'm half deaf. Single nucleotide polymorphism. Why would you like lots of those? Because then the variation in the genes. Variation in the genes might translate into variations in the phenotype, right? If everybody's got the same gene, is it likely to have a differential phenotype in its expression? Yeah, probably not. All right, so we looked where the SNP density was high, and we also then bounced ourselves into the UC Santa Cruz genome browser from this, and I'll be talking about that ere long. Okay. And, in fact, right now. So the other thing I wanted you guys to see was a whole bunch of bioinformatic tools, and that was one of the focus of this module. These are tools that even as I speak, real deal scientists, biologists are using right now to figure out questions like we address. You know, what's affecting a phenotype? How are genes different between a disease state and a non-disease state? Things like this. So these tools are out there. You may recall when I first started this unit, I talked about Gregor Mendel and how his paper essentially got lost for 40 years. People forgot about it. We're generating piles of data right now. We want to keep access to it, and some far-sighted people have created these bioinformatic tools so that we can keep access to that data and use it in a very accessible fashion. So you guys got to see some of these. One of them was this QTL analysis, which is out on the web for free, courtesy of the University of Tennessee and people there. What we then jumped into out of that web QTL was the UC Santa Cruz genome browser, and what that told us was on our chromosome, these by the way are some data I made up, but this is on chromosome 6. For this little patch of chromosome 6, this listed all the genes that are known, and what you guys did was essentially data mine and go through those genes and find one that was highly expressed. Okay? And when I did it on my own and had my own made-up data, I found one GABA receptor A protein, and I found one that was highly expressed in olfactory bulb, just like you did. Okay. We also talked about the fact that this expression data came out of something called DNA microarrays or gene chips, and we talked a little bit about those, how those are made. We used fluorescent cDNAs. What it gives us is the pattern of gene expression across different conditions. And so I believe the way that they got that relative expression was to look at cDNAs that came out of an olfactory bulb, say, relative to a standard bunch of genes, okay, a standard mix of genes. It allows us to look at thousands of genes at once. The downside, at this juncture at least, is it doesn't give cell-by-cell resolution. You have to look at region by region. And I wanted you to hear about these. These are now widely used in research. They're also popping up in the clinics. As I said, people are looking at different kinds of breast cancer tumors to see the pattern of genes that they may display that helps the clinicians figure out the treatments that they are going to prescribe. Okay, and then we jumped from the UC Santa Cruz genome browser into the Allen Brain Atlas. And what that is, they have all these people up there in Seattle doing in situ hybridizations for gene after gene after gene after gene. I mean, they literally have just armies of technicians. And so they have some in situ data. So we can, once we found our gene that was highly expressed in the olfactory bulb, then one of the questions you might want to ask is, what cell layers is it expressed in? And so this Allen Brain Atlas will tell you that. You can go there, and if that gene happens to be in their collection, lots of them are by now, you can see here's the in situ, and this is a gene that was pretty ubiquitously expressed, but you can see here in my olfactory bulb, it's really expressed in that layer. This was an in situ that was done with digoxygenin, so you get this reaction product that's kind of this deep purple when there's labeling. And what I also had you do was look up the reference atlas, so then if you saw a pattern like that, you could look up the reference atlas that they've also created and find out, okay, what's the name of that cell layer? All right, so that's available to you too as a bioinformatic tool. Then I had you go to Entree Gene. Once you had your gene that was highly expressed and you knew where it was expressed, this should have been something that looked fairly familiar to you from the Bolter and Weinmaster unit. We finally ended up at a page where you could find out the actual sequence of that gene and also the coding sequence for that gene. So if you wanted to go in and say PCR part of it or something like that, or do some other manipulation with it, you could know that's sequence. And then also out of this UC Santa Cruz genome browser, we went to PubMed, which is your tax dollar at work, and it gave you some articles about that gene. I had you find one, and hopefully you're reading it, and you're going to tell me all about that article. All right? Okay, so those are all bioinformatic tools. Just to summarize, so what WebQTL did, it performs that QTL analysis for us and gives us the likelihood ratio statistic as a function of markers on chromosomes. UC Santa Cruz Genome Browser tells us the known genes over a given region of chromosome. Probably with the mouse we know all the genes, and gives the relative expression levels of genes. Allen Brain Atlas then gave us the in situ hybridization, where you could find out cell layers and cells in which the gene was expressed. Entree Gene gave us the sequence, and PubMed gave us some more information. So again, these are all tools that even as I speak, people are using, and I wanted you to see them, okay? Because when you get out of here, if you get a job in research, you're going to be using these things, very likely. And then my last objective was to review and introduce some molecular techniques. We did in the Bolter Weinmaster module, PCR technique. So this should have been quite familiar to you. So that gave, PCR is used to find out, again, the difference for these different recombinant inbred strains if they have the B or the D form. And again, just I can't emphasize this enough, this is the first F2 generation, right? So after they've been inbred, they're not going to have, you're not going to see any of these heterozygotes. They're going one way or the other. But anyway, the way those markers are discovered is someone goes through and PCRs up little hunks of DNA and sees if your F0s are different in that little hunk of DNA. And so it's a technique that's allowed us to put markers at different places in chromosomes so we could track, did that come from the F0B strain or F0D strain or some other cross for that matter. I've already talked a little bit about gene chips, and so I'm going to just skip over this slide. But again, they are in research. My colleagues are using, we have one now for the zebrafinch, so we're trying to figure out if boy zebrafinch genes and girl zebrafinch genes are differential relative to a mix of genes, so they're used widely in research and also in the clinic. Do you guys hear about in situ hybridization in other classes? Did you Neuroscience? What other classes do you hear about it? 101C? They're doing a good job there, Joe. Okay, so just to review, what in situ hybridization does is it allows us to find out which cells express a gene by probing for the mRNA. And why would we care about mRNA? It's on its way, hopefully, to making a protein, right? And the probes to that mRNA are usually made out of RNA. The ones I've done are RNA. Or you can make them out of DNA also. And we make probes that are what we call antisense probes. So the mRNA we call the sense strand because it's coding for the protein. So the one that is its complement we call by the antisense strand. So you make an antisense probe. And you also, when you do that, make a sense strand, and you use that for your control. The probes are labeled, the Allen Brain Atlas that we saw earlier, the probes are labeled with something called deoxygenin, which makes a color reaction. You could label them with radioactivity, which people don't like to use very much because it's too dangerous, but it does still get used. The probes are then, when you splash them on the tissue, they'll hybridize with the specific mRNAs, hopefully, being made in a cell, so you can find out cell by cell what cells are producing that mRNA or expressing that gene. The downside is, at this juncture, you can probably only look at a couple of genes at a time, okay? So you can't look at thousands and thousands. Just no way to do it right at the moment. No one's been clever enough to figure that out. Maybe one of you will. Okay. And again, what you get, this is a gene that was pretty ubiquitously expressed. You can see it up here in the olfactory bulbs. What's this, by the way? You neuroanatomy fans. Say it loud. Hippocampus. How about here? Cerebellum. So it's expressed in all these really heavily. For this particular, this was the GABA A receptor protein, so that's no big surprise. That's pretty ubiquitous, right? Yeah. All right. But at any rate, you found a gene that was hopefully highly expressed in olfactory bulbs and tracked down what cell layer it was in. Okay. And I can see that my last slide got cut off. What happened to it? Yep. All right, let me see if I can find it. All right, the last thing that I need to tell you is about things that are coming up. And the other thing that I emailed you besides, nope, I don't have it, besides how to get some extra credit is next week we're going to start the next unit, which is SWIMMY, which is virtual electrophysiology. So in that email that I sent to you, I sent you a URL where you can go and download the manual. You might want a hard copy. It's probably more efficient if you have a hard copy. All right, I know it's a lot of pages, but probably you'll do better. If you want to download the program, it's also there to play with. It only runs on the Windows platform. And as an added attraction, Dr. Krasny will be distributing a quiz next Monday. Okay, so you probably want to come to lecture. All right, and don't forget, next week, start a lab. Your write-up for this module is due. Rick's going to have office hours starting momentarily, and also on Monday. I'm going to have office hours on Monday to help you with that. And don't forget the turn-in copy as well as the hard copy. Yeah? No. It's a make-you-think quiz about electrophysiology. Okay? All right. Thank you for your kind attention.